So I'd like to thank the organizers for inviting me to this meeting. It's not the kind of meeting that I usually go to. Now, I think we've become so specialized that we tend to go to meetings that are just in our area, and yet, sorry. Yeah, I think this is a good thing. I think we need a sort of get a better perspective on other areas of research that I think can have a really big impact on what we're doing. So thank you, Ron, for that good introduction. I don't need to show any of the basic immunology, which obviously is critical for understanding.
You can do it with the arrow on the keyboard.
The arrow on the keyboard?
I don't know how to get it back.
Okay.
And then use the right button to advance.
Okay. So, as I'm sure all you are aware, adoptive T-cell-based immunotherapies have been very effective, primarily in melanoma. And this has been an area that's been studied for decades now. I think people perhaps don't appreciate that this has been something that has been carried on by the surgery branch as well as other groups for many, many years. But as Ron had said, with the recent advent of checkpoint blockade inhibitors, as well as immunotherapy with CD19 chimeric antigen receptors, I think it's become something that people are certainly a lot more aware of. The basic approaches can be summarized in this slide. Basically, you can isolate tumor-infiltrating lymphocytes, grow them in vitro. It's a relatively straightforward process for melanoma, and actually we're now using this in other cancers as well. And then expand those cells and give them back to the patient. Generally, we administer IL-2. We also use a preconditioning regimen, which can include chemotherapy as well as total body irradiation. This is to provide an environment for the cells to expand, as well as, we think, to eliminate negative factors such as regulatory T cells, which are present in high numbers in tumors. And so we think this provides an opportunity to modulate the host environment that's really critical, I think, for some of our therapies. The other approach I won't really talk about is, but Ron introduced this, was the genetic engineering approach to introduce T-cell receptors or chimeric antigen receptors into a patient's own autologous T-cells. This is a summary of a trial involving 93 patients who either received no total body irradiation or total body irradiation, and we see good response rates, about 50% response rates. In subsequent trials, we haven't seen much of an improvement with total body irradiation, so there's an important caveat here that when you're dealing with small trials, with small numbers of patients, these effects do not always reproduce. But what is reproducible is the long duration of responses. All of these are the responses in months, with a plus indicating ongoing responses. So patients are now responding, many of them 5, 10 years after their therapy. And these are complete responders. So of all of the patients in this trial, you can see with the Kaplan-Meier curve, this is the overall survival. Patients treated on these trials, all of the patients in the non-myeloablative chemotherapy trial, and then they're broken up into patients that either didn't receive total body irradiation or received total body radiation. But these are long-term survivals, as I said, out beyond 5 years for many of these patients. So these can be very effective. Another important point is that patients who do have a complete response generally maintain those responses for many, many years. One patient out of 20 who had a complete response recurred, but all of the other patients have these long, ongoing, complete responses. So what I'm going to talk about is antigen specificity, because clearly that can have a major impact, we believe, on the response, although other things that I've studied also have an impact. We showed several years ago that the telomere length of the administered cells was important, and their in vivo persistence following transfer was important. Now, this makes inherent sense. If you have longer telomeres, the cells have a higher proliferative potential and presumably are able to maintain a state of, maintain the patient tumor-free for longer periods of time. But antigen specificity clearly may be related to clinical response, although this isn't something that's so easy to determine because we're administering bulk polyclonal populations of cells. These are all of the cells that have grown in vitro from a patient's tumor. But clearly this is something that I think bears a lot of study. So if antigen specificity is important, what are the important characteristics? What are the distinguishing characteristics of those tumor rejection antigens, and how can we translate these into developing more effective therapies? As Ron mentioned, we don't know what the antigens are that are being targeted by in the patients that receive checkpoint blockades. But clearly, I think even in those patients, having a better understanding of what these tumor rejection antigens are might allow us to improve those therapies. Otherwise, we're working kind of blind in a sense. So what I'm going to focus on here is using next generation sequencing methods to really identify the mutated antigens that are recognized by T cells. Now, I don't have time to go into a lot of the background that basically led us to believe that mutated antigens were more important and maybe the most potent tumor rejection antigens that are being recognized by our TIL. But suffice it to say that a lot of work has been done on characterizing TIL in terms of their ability to recognize shared antigens, such as the melanocyte differentiation antigens MART1 and GP100. These are pretty easy to identify because they're very common epitopes, particularly in HLA-A2 patients. And so it's easy to identify those cells. So we had administered T cell receptor transduced cells to patients that were highly potent, in vitro, capable of killing cells, making large amounts of cytokines. But we had very disappointing results. And in addition, we had autoimmunity. We had eye, ear, skin toxicity. So these cells were potent. They were capable of recognizing even the normal cells, the melanocytes that are present in our skin as well as in those other tissues. And yet the response rate was very disappointing. So it's hard to sort of reconcile those findings with the thought that TIL are targeting those antigens. Because if TIL are recognizing those antigens, why don't we get those same kinds of autoimmune reactions in patients who receive the TIL? So this is what sort of led us to thinking that it's not those common shared antigens that are the primary targets, but actually the neoantigens, the antigens that are derived from tumor-specific mutations that may be the basis of the really potent regression that we see in many of these patients. Again, it's just a correlation. Certainly, it's hard to prove causation in any kind of human trials. So this slide really summarizes the approach, though, that our group has taken, basically to utilize whole exome sequencing and actually, in recent studies, to use RNAseq to identify the mutations, and then to design and synthesize what are called mutated tandem minigenes. I'm going to abbreviate these as TMGs. These are basically just short synthetic DNA fragments that can be concatenated into vectors and then be used to either transfect or transduce autologous cells. If you use this approach and use the patient's own dendritic cells, for example, you can now get antigen presentation of all of the epitopes that are possibly presented on that patient's own MHC molecules. Alternatively, you can use short or long peptides. By long peptides, I mean 25 mers that encompass the mutation, but that can be processed by dendritic cells, or short peptides that were predicted to bind to particular HLA alleles for that patient, and again, use autologous cells to present those peptides. So I'm going to discuss some of those approaches and show the results of some studies we've done there. In addition, at the same time, we can use different methods to derive T cells. We can either digest the tumor, and so we get a bulk population, and then either grow those cells in vitro as bulk cultures or to sort them into different populations. Obviously, there are CD8, CD4 positive T cells, class 1 and class 2 restricted T cells in these tumors. Those cells also express markers like PD-1, which can be used to sort the cells that are tumor reactive because those cells have upregulated the inhibitory marker PD-1 in the presence of the cells, in the presence of tumor cells, despite the fact that they're not causing regression of the tumor, and that's part of the natural balance. The tumors are upregulating ligands for these inhibitory molecules that are being targeted in these recent trials. We can then expand those cells and then identify the cells that recognize either these transfected genes or peptides that we've generated. And we can then derive cultures that we can be used to treat patients using a selection approach that I'll describe a little bit later. But an important part of this is, of course, sequencing the tumor cell and normal cell DNA, either for identifying T cell receptors that we can then use or for identifying the mutations that we can potentially target with our therapies. So this is a little busy slide, but I wanted to sort of summarize all the approaches that we're taking now to utilize this information to develop these therapies.
How deep do you sequence it?
How deep? Generally, exome sequencing, we have been sequencing about 100, 150x, which may not be sufficient for all of our tumors. And I'll actually show some data a little bit later that impacts on that. So here's an example of using this sequencing approach to identify candidate peptides. Now, as Ron mentioned, MHC molecules bind peptides, but those peptides have certain characteristic residues that allow them to bind to a particular MHC allele. So you can identify the characteristics of peptides that are at least predicted to bind to a patient's own HLA alleles, and then synthesize those peptides, and then test the ability of TIL from those patients to recognize targets that are pulsed with those peptides. And here's an example of the use of that approach. These peptides were predicted to bind to HLA-A1, which is one of the HLA restriction elements expressed by that patient's tumor. And these are the peptides that were predicted with the mutation position here underlined. And then in red, what are shown are the peptides that are actually recognized by that patient's cell. So we just simply do a straightforward co-culture assay where we pulse those peptides on target cells and look for the ability of T cells from that patient to produce interferon gamma. And here 3 peptides are recognized. 2 of them are actually overlapping peptides. So essentially they're the same epitope. One is a 9 amino acid peptide. The other is a 10 amino acid peptide derived from PPP1R3B. This is one of the regulatory components of a protein phosphorylase. And then this is PLEC-HM2. Again, the function of these genes is not something that we investigate. Obviously, some of these mutated antigens could have important functional aspects to their mutations, but it's not something that we have investigated any further. In any case, we can show that T cells recognize these particular mutated epitopes. And so we now know potentially the important antigen targets for this patient's TIL. And interestingly, for this patient, we never identified any shared antigens. So we tried to see if there were other antigens that might be recognized, but we couldn't identify them. Another example of this approach is to use HLA tetramers. So you can generate peptide libraries using a very clever system that Ton Schumacher developed, where you have a peptide that is UV sensitive. You can incubate that MHC peptide complex with ultraviolet light. That peptide is degraded by the light, and if you add another peptide that's capable of binding that MHC molecule, you can then rescue that MHC molecule. That MHC molecule is unstable without a peptide in the binding groove, but if you add another peptide, you can then allow that to to maintain its native conformation, and then use streptavidin to multimerize it, and then these can be generated, large libraries can be generated very quickly using this approach. And you can then screen a patient's cells. And an example of how this was done is shown here, where Cyril Cohen, senior investigator who was working with my group for the past year, isolated cells from a fresh tumor. So this is a tumor that's just been digested with enzymes and then stained with a particular tetramer that's now actually been labeled with 2 fluorophores to allow it to be more easily picked up. And only a small percentage of the cells here are bound, but nevertheless we can show that those cells can be isolated and then expanded, and they're specific for this particular mutated epitope. Here's another example where there's a 1 and 0.2% of the T cells recognize these two different tetrameric complexes. One of them, actually, interestingly enough, is from ERBB2, a protein certainly that everybody here is familiar with. Another example of the use of this approach is to sort them from peripheral blood. So even prior to therapy, these cells exist in the peripheral blood. They exist in most cases at very, very low levels. But in this case, 0.2% of the cells in peripheral blood were bound by this particular tetramer from TRP12. This is actually the highest percentage of any of these tetramer-positive cells for any of the epitopes that we've looked at. Generally, the percentage of cells in peripheral blood are in the sub 0.1%, even less than 0.01%. But you can sort phereses from the patient's blood, isolate those cells, and expand them in vitro several thousand-fold, and generate a population of tumor-specific cells. So this is, I think, an exciting approach because you don't need the tumor. You can just take the patient's blood, isolate these cells, expand them, and potentially give them back to the patient. Yes.
What would be the percentage in the T cells in the tumor? What's the comparison between peripheral?
Yeah, the question was what was the percentage in the tumor. In this case, I don't recall. It's a higher percentage, but yeah, generally when we have compared these, it's a significantly higher percentage in the tumor.
10-fold higher?
Honestly, I don't recall the numbers for this particular antigen, but generally they're much higher. For these cells, I know it's definitely much higher. I mean, there are 1% of the cells reactive with this collagen 18A1 peptide. In the peripheral blood, it's probably 100-fold higher than in the peripheral blood. So this slide summarizes the results of our screening approaches over the years that have identified mutated antigens, and to be sure, use of conventional screening methods such as cDNA library screening of methods have resulted in identifying, on this slide, 11 mutated antigens that are recognized by melanoma TIL. But over on the right, you can see a list of over 40 antigens that have been identified using whole exome sequencing approaches. And the important point here is that these antigens were identified by a fairly laborious screening process over a period of 2 decades. These antigens were identified in less than 2 years. So these techniques have allowed us to streamline and develop an approach that can be used to very rapidly identify mutated antigens. You'll also notice down in the bottom here, I've listed colon and rectal and cholangiocarcinoma samples. So we've now identified T cells that actually recognize GI cancers fairly reproducibly, which is a somewhat surprising, but I think very significant point. So on this slide, I summarize the results of our recent experiments to identify mutated epitopes using either this Tandem Minigene approach, our mutated tetramers, or in some cases just use of a conventional cDNA library approach. And what I'm showing you on this column is the number of mutated epitopes that this patient's T cells recognize, and the number of non-synonymous mutations. And as you can imagine, the number of T cell epitopes that are recognized is dependent on the number of non-synonymous mutations that are present. Now in this list, most of these patients are responders. And I didn't put the response data in here because I didn't want to emphasize or make the point that response is related to the number of mutations, although obviously if you have this kind of data, you could potentially look at that. And now this presumably will have some impact on the therapy because, for example, this patient's TIL recognizes 10 epitopes, and that's 10 epitopes that we've been able to identify. There's probably more that we haven't been able to identify. But in terms of the specific issues of this conference, which is to look at genetic diversity, certainly tumors that have large numbers of mutations are going to presumably have larger numbers of targets. So if you have a lot of genetic diversity, there's more targets that you can potentially attack in that tumor with that patient's T cells. I will say, though, that these two patients are extremely good long-term responders. So, but I don't want to sort of overemphasize that point because patients that have relatively low numbers of mutations, some of these patients down toward the bottom of the list are also good responders to TIL therapy. The other reason I didn't want to show that is that actually some of these patients' TIL have only been screened using one approach, using, for example, these three patients here. We only used the mutated Tetramer approach. We haven't gotten to the point of using other methods to screen. So we may be able to identify additional antigens for these patients. Nevertheless, this is the kind of landscape of tumors and the sort of range of non-synonymous somatic mutations that we can potentially target in these tumors. Now this slide shows the results of studies of GI cancers, mostly carried out by Eric Tran in our group. And as you can see, 5 out of the 8 patients that he screened actually contain mutated epitopes that were identified. These were all identified using this Tandem Minigenes screening approach. And the number of mutations present in these tumors is actually relatively low. As you can see, one patient here had only 46 non-synonymous somatic mutations. So even with relatively low numbers of mutations, it's possible to identify T cells that recognize at least one of those mutations. And then these are actually ongoing experiments. It's possible we'll identify epitopes that are recognized by all of these patients' TIL. But another important point to make is that colon cancers do not respond or don't appear to respond to these checkpoint therapies. So the response rates, the relationship between response rate and mutation rate isn't clear, but nevertheless, PD-1 therapies, at least to this point, have not been successful in colon cancer. And they will not be successful in I think, a lot of cancers. Whether that's related to the mutation rate or not, it's too early to tell. But it'll be interesting to see if there is some kind of correlation between the success of that therapy. But the reason that it fails is not because T cells from GI cancers can't recognize their tumor, because these are examples of T cells where we have identified epitopes that are recognized by the T cells. So undoubtedly, there's many layers of control of these tumors that are overlaid on just the immune response. So it's obviously much more complicated than that. But I think we're just starting to sort of understand the nature of the T cell response to these cancers and how that is impacted by PD-1 therapies. Now I want to go through just very briefly an example of how this screening approach was used to direct a therapy for a patient. And this was carried out by Eric Tran and recently described in a science paper. It was published in May. This patient was had metastatic cholangiocarcinoma, had been extensively treated with chemotherapy, and he was treated with TIL and had tumor regression. It was a relatively short regression and then stable disease for a period of about 13 months. Now, what you can see here is the lung CT scans where you can see these lung tumors not completely regressing, but certainly there was a substantial response here. The liver lesions regressed and actually continued to slowly shrink, but the lung lesions then began to recur. So you can see here this in the open triangles, the response in the lung. An important point here is we treated this patient with about 40 billion T cells. So what was done was to generate constructs that encode mini-genes that cover the mutations that this patient's T cell recognized. Initially, what Eric did was looked at 26 of the mutations. Subsequent analysis has shown that there are probably a few more mutations that could be targeted. And so these mini-genes are basically just strung together in tandem constructs. Generally, we use between 10 and 20 of these mini-genes in a single construct. And then we in vitro transcribe RNA from that construct and transfect into an autologous antigen-presenting cell. So now all of the potential peptides, either class 1 or class 2, that can be generated with that patient's MHC are present on the surface of that dendritic cell. And then those are simply co-cultured with T cells. And what he found was that one of these tandem mini-gene constructs encoded an antigen that was recognized, as shown in this ELISpot assay, where each of these spots is a T cell that's secreting interferon gamma. And here's a FACS plot showing upregulation of a co-stimulatory molecule, OX40, on the CD4 cells that were stimulated with this transfected mini-gene.
Can I just ask,
Yeah.
if those are mutations, none of them are structural alterations?
Those are all non-synonymous mutations.
Yep.
Right. With such a low number of mutations, generally we find very few indels. I mean, obviously there's some colon cancers have genetic instabilities. You have Lynch syndrome patients where you have mismatch repair defects, and those tumors have very high numbers of mutations and many indels. But translocations, yeah, that's something that we really haven't begun to look at, but it's actually much harder to identify those, but presumably they're relatively few. They could serve as targets, though. So I guess that's something that we need to really look into a little bit further. But primarily we've targeted non-synonymous somatic mutations.
And when you look at actionable items from Foundation Medicine,
Yeah.
the structural alterations are actually something like 3 to 2 over mutations. So the structural alterations are actually more actionable in their report.
Something we probably should look at. Again, I think that the kind of analysis that's required to sort of determine those, and don't forget, you have to determine the precise breakpoint and the precise sequence. You can't just simply look for an alteration. We require the actual amino acid sequence at that site. So I think that this is a case where the bioinformatics is maybe a little bit behind, because of course when you're predicting mutations, you have to map it to genomic loci. If there's a breakpoint, now the sequencing analysis programs will often throw those out, will discard those. So it's a bioinformatics issue, and I would love the bioinformaticians to help us out with that. Because
just trying to help, not trying to criticize.
Right. No, I understand. That's perfectly valid.
We found that with paired-in sequencing, it's actually very easy to find the translocations, and then that you can hone in on the breakpoints. But you're right that most of at least the early software is hard to find, which we're out. But,
Yeah.
but it's quite doable.
Really? Okay. Yeah, we probably need to look into that a little bit more, and I need to talk to our bioinformatics people about that. So here's an example then of how he then went on to identify the particular antigen, and it was ERBB2 interacting protein. This is a protein that binds to and regulates the function of ERBB2. And when he reverted the mini-genes in this construct back to the wild type when he reverted this particular coding region to the wild type, he showed that there was no recognition now of the transfected target. So that was the actual antigen that was being recognized. But critically, what we did when this patient's tumor began to progress after the first treatment was we went back and looked at the original cultures that were used to derive the what's called the infusion bag, the cells that were actually administered to that patient. And what we saw was that only about a quarter of the cells express this particular T cell receptor, which he actually showed recognized the ERBB2IP. This was identified using a V-beta antibody, which can be used to track individual cells, or at least subpopulations of cells. And so we noticed that one of the fragments was very highly enriched in cells that recognized this antigen, and that was selected for retreatment. And this patient now received a large number of cells, actually 10 times as many cells, that recognized that ERBB2 interacting protein. And now what you can see is that the tumors have begun to regress again in the lung and in the liver. They're continuing to regress. And now 8 months beyond therapy, the tumors are still shrinking. So we're hopeful that we're going to achieve a long-term response and may even achieve a complete response if these cells continue to act and cause tumor regression. So I think this is a powerful example of how you can use this information to treat a patient's cancer. And this is essentially a monotherapy. So I think that even though we've talked a lot about tumor heterogeneity, certainly there are certain antigens that can be targeted in the tumor and lead to potentially complete responses. But getting back to the tumor heterogeneity issue, what do we really mean? And we've talked a lot about this, but it can mean disparate somatic mutations in tumor cell clones or clades in a particular patient, disparate epigenetic states of those cells in clades or even different regions of the tumor. But I think also important is differing types and degrees of normal cell infiltrates, which Carlo has mentioned, differing levels of apoptosis as well. And these factors have important practical implications, I think, that may fashion or limit our therapeutic options. I'm going to show you two examples this is H&E staining of some OCT blocks. So these are frozen sections that were examined prior to the extraction of DNA. We do H&E sections basically to determine the level of tumor in those OCT blocks. And this is an example of what I would call a fairly homogeneous tumor. There's some stroma in there, but clearly a fairly homogeneous population that was judged by the pathologist to be essentially 90% tumor. Here's an example of the other extreme, where there's a lot of stroma, but a lot of apoptosis, infiltration by other cells. The tumor cells have these large nuclei, but obviously there are many cells with relatively small nuclei. And this was judged to be only about 30 to 40% tumor. So we have these samples that we get, and it's not easy to separate the normal from the tumor here. So we basically just have to take what we get and carry out the exome sequencing on these samples. And the problem is, of course, with a lot of normal infiltration, we're going to have problems with identifying all of the somatic mutation. We're going to be diluting out all of the variants that we see. Now, basically, this is the first patient, and what I'm showing you here is an example of the kind of approach that we're using to validate as well as extend our results of whole exome sequencing. Here, I'm showing you the whole exome variant allele frequency of the fresh tumor, but also RNAseq analysis of that fresh tumor sample. So when you look, you can see that there's a fairly good linear correlation between those variant allele frequencies. Some of these are very frequent. Essentially, we have a loss of heterozygosity, so 100% of the calls at that particular position are variant. And there's a wide range in variant allele frequency. So this undoubtedly reflects the tumor heterogeneity. Some of these variants are present at only relatively low frequencies, but there's a wide range. Here's an example then of the comparison that we carried out by doing whole exome sequencing and comparing it with RNA-seq, but now of a short-term tissue culture cell line that we derived from that patient. And we see a very similar pattern. Again, a nice linear relationship between the mutations that we identified by whole exome sequencing and by RNA-seq analysis. In melanoma, obviously, we have the advantage that we can often generate short-term cultured lines. Now, this is the sample, though, from that patient where there was extensive necrosis and only a relatively low purity tumor. And as you can see, when you carry out whole exome sequencing, all right, you have essentially no variants that have higher than about a 20 or 30% frequency. But when you carry out RNA-seq analysis, you actually have a fairly large number of these variants that are called at fairly high frequency. So the point I want to make here is that tumor heterogeneity, at least as reflected in the infiltration of normal cells, has a major impact on the results that you get, particularly when you carry out whole exome sequencing. Many of these would not be called as variants by whole exome sequencing because the frequency would be so low, you would basically want to ignore those and throw those out. And not only from the point of view of the statistical analysis, which once you get down to very low frequencies of 5 to 10% becomes a problem. So you have read count issues, you have low frequencies. You probably wouldn't want to call a lot of these based on the whole exome sequencing results. However, with RNA-seq, you can actually see that you could probably make a lot of these calls with high degree of confidence because the frequencies are much higher. And in addition, I'm not showing you this here, but basically for many of these variants, we also have a very high depth of read because many of these are only transcripts that are expressed in the tumor and not in the normal infiltrating cells. So I think, again, this is a practical issue, but something I think we need to think about in terms of how we analyze these samples. Imposed on top of that, however, is the fact that exome capture is a relatively inefficient process. So people that do this a lot know that you actually capture with a widely varying degree of efficiency individual exons, all right? And some exons aren't captured at all. It's been estimated that about a quarter to a third of all exomes basically are not captured, at least by the current exome capture kits, despite what Illumina and all of the other companies will tell you. So you have this relatively low efficiency of capturing a lot of the exons. So I think we're underestimating, perhaps severely in some cases, the number of mutations, the number of mutations in these tumors when we carry out whole exome sequencing. Now, the reason I can say confidently that a lot of these are actually correct calls, even though the whole exome frequencies are very low, is that we had the advantage of having a tissue culture cell line from this patient's tumor as well. So now when we go back and look at that data again, and look at the RNA-seq analysis by whole exome versus RNA-seq, we can see that very nice linear correlation again. So, and if you, again, it's difficult to show this on a single slide in 2 dimensions, but basically many of these variants, map back to the variants that we found in fresh tumor. So it's a cautionary tale that when you're actually carrying out analysis on fresh tumors and samples that we get from patients, I think in many cases we may be missing a lot of information. Yes?
On the upper right corner,
Yeah.
the RNA-seq and whole exome has been performed on the same sample, right?
Okay, in this case, we have not actually performed RNA-seq on the OCT block because we find that the integrity of the RNA is lower. So we have what we term snap freezes. So there's simply areas of the tumor that are close by the area of the OCT block that we've taken, okay?
The tumor purity, is it the same?
We don't actually run H&E sections on that, so but I understand your question. Okay, to really flesh this out, what you would have to do is whole exome sequencing of that sample as well. All right, along with the RNA-seq analysis. I mean, and we're in the process of doing this, but again, it's a lot of work to do whole exome sequencing of the snap freezes of the OCT, and now we have cell line data as well. But we're starting to integrate this to try to understand how that's impacted. Obviously, tumor heterogeneity can impact that as well. But I will tell you in this case that we don't have any problem aligning these reads to the reads that we get from the fresh tumor. So a tissue culture cell line, which was generated actually from a fresh tumor digest, so that's an independent sample, also contains the same mutations that we find in either the snap freeze or the OCT block. So there certainly is a lot of diversity, but many of these mutation calls, particularly the high frequency calls, can be made from any of those samples.
Yeah.
Okay?
But assuming the tumor purity is the same, you would only expect to see this if the mutated variant is also higher expressed in the tumor than in the normal.
Okay, but don't forget, RNA-seq analysis analyzes transcripts, right? Some of which at least are expressed only by the tumor, or are going to be expressed at higher levels in the tumor than in the normal. Now, a housekeeping gene obviously would be expressed at high levels in both. Right? So we haven't gone back and tried to sort of sort all that out. But I think at least for many of those genes, they're not expressed in the normal tissue. Now, the next question might be, well, what about a normal transcriptome, right? That's a lot more difficult. We could do that, I guess, potentially, but it's just not something that we've really embarked on. I think that's a good question, but I think it's something that we may do. It's something that we may do. But obviously, there are different cell types. In the immune infiltrates, there's stroma. So the presumption here is that many of these transcripts, though, are expressed either exclusively by the tumor or expressed at much higher levels in the tumor than in the normal. Basically, I just want to finish up here on this one tumor, a further example of sort of pairing or using the RNA-seq analysis and whole exome sequencing to identify mutated antigens. And those of you who carry out sequence analysis will notice that we've actually used a very low minimum variant read call of only 3 variants, so 3 mutated sequences as our cutoff here, and very low allele frequencies. But I think that by merging the 2 data sets, we can get much more reliable information, and this is an example of the combined analysis. This is exome variant allele frequencies, and as you can see, some of these are fairly low. In addition, the number of reads that we get is very low too. We only got 2 variant calls from the exome data. So that's something that most people would not include in their sequence analysis because statistically having just 2 variants is likely to be an artifact. However, we have RNA-seq analysis that's able to make confident calls, confident calls, because we now have 22 and 68 variant calls. So by merging this data, we can actually develop a much more robust set of exome sequence for our analysis. And this is the last slide. Basically, I just want to say that in this patient here illustrates another issue that's relevant to tumor heterogeneity. This is a metastatic melanoma patient that had metastases in the pelvis, omentum, the brain, received adoptive TIL transfer. These TIL were derived from the right hip and back area and received these cells in March 2006. All disease regressed with the exception of a couple of small lesions, one of which was resected and was found to contain residual tumor. A short-term cell line was generated from this omental mass, and whole exome sequencing was carried out on both of these lesions now. So these are completely independent, separate lesions. So the initial analysis of tumor 2556 led to the identification of MYH14 and RAC1 as T-cell antigens. Now, RAC1 is a driver mutation in melanoma. It's present in about 10% of melanomas, and there's a hot spot. There's a single amino acid change at position 29, single change from proline to serine that's identified in about 10% of melanomas. So this is a potential target that actually is not restricted to this particular patient. This potentially could be used in a much larger number of patients because of its prevalence. Now, the point I wanted to make here, though, was that when we did RNAseq analysis of the patient's original tumor and the recurrent tumor, we found exactly the same level of expression of both of these mutations. So you could look at this as good news and bad news, okay? In a sense, the tumor has not changed. The tumor has not evolved or has not developed resistance by eliminating or downregulating these mutated antigens. It's still there in the lesions. It's an example, I think, though, that shows that our therapies still aren't potent enough. They're not potent enough to lead to immune selection. And I think that's an important point to make. We're talking about the immune system as driving diversity, potentially leading to the outgrowth of variants that don't express these mutations. And yet here's an example where two different lesions had the same mutation even after treatment. We were able to resect this lesion, though, and the patient is now disease-free. So in this case, because of the limited expression in only a couple of additional lesions, the patient is doing fine. But I think, again, it makes an important point, and perhaps part of the reason for that is that, in fact, in order to develop resistance to the therapy, the tumor would not have to eliminate one, but two of these. Okay? And so that's putting a lot more selective pressure. All right? You would have to basically inactivate both of these mutations by some mechanism. So I think it points to the fact that diverse TIL are potent or can be very potent because of their ability to recognize these multiple mutations. Yeah.
Doesn't it suggest that it's finding some other form of resistance, some other way of avoiding immune?
Yeah, I mean, it absolutely says that, I believe, it says that despite the fact that these could be targeted, again, in a human trial, it's impossible to show causation as opposed to just correlation. It suggests, you're right, that there are other mechanisms in that lesion that allowed it to resist the therapy. And that's where we need, I think, combination therapies. We probably need anti-PD-1. We need these other checkpoint inhibitors to really develop even more effective adoptive immunotherapies. Because if PD-1, that axis was very active in that particular lesion, that might actually have been the reason why that particular lesion didn't respond. Yes?
T cells also have to have equal access to all your metastatic sites.
Right.
So when you resected these, did you look at the infiltrating lymphocytes in those sites to see if there's any difference?
Yeah, I mean, we could, the question was, do T cells have equal access to all of these sites? And that's another interesting aspect to this whole thing. We could now go back and do that because we can track the cells that actually have specific receptors against those and look for those. We haven't done it yet, but I think that would be an interesting question to see if, in fact, those cells were even there in the tumor.
I appreciate you bringing up the point about, lower tumor purity radically affects how many mutations you're able to call. And I'm curious, do you think that the correlation between mutation burden and response to immunotherapy may just simply be correlated to the level of infiltrating normal cells?
That's a tough question. I mean, it's the level of infiltrating normal cells. I don't think in general, if you just want to say sort of on a semi-quantitative basis, I don't think that varies. Well, it's hard to say. It varies from melanoma to melanoma. I mean, obviously, it's not a homogeneous thing. One way we have of looking at that is Elena Gross, who published a paper in JCI recently, showed that PD-1 can be used as a marker to enrich reactive cells. And so you can you can sort of use that as a gauge, at least of the percentage of cells in a tumor that recognize that targets in that tumor. But she hasn't been able to show any correlation between the percentage of cells that recognize mutated antigens and subsequent response. So those kind of quantitative things, I think, are going to be difficult, but they're worth looking at in the future. And maybe we just haven't looked at enough patients. It's an interesting question. Yeah. Okay.
Yeah, great. I'm glad to see the trials actually applied to the patients.
Right.
And one question is that you have 2 doses, right? In your later example, that you put the first dose and then you realize you add more in the second phase. How do you determine those doses for how much is enough? And
that's a sort of a practical issue. I mean, we have limited resources. We can expand cells. We use OKT3 and feeder cells, but to expand, to those 50, 100 billion cells, it obviously takes a lot of feeders. And so we basically, on a patient-by-patient basis, we use a fairly standard protocol, but we can't determine ahead of time how much those cells will expand, for example. So we'll take 50 T150 flasks, harvest the cells from those flasks, and that's what we get. So obviously we can ramp it up for an individual patient, and in this particular patient, since we felt like the number of cells perhaps was too limited, we tried hard to generate more cells. But that's a practical issue. That's I mean, it's hard to say. We can't say there's a certain number that's needed for therapy.
Okay.
But we have general sort of protocols that we follow. Yeah.
Paul, back to the tumor heterogeneity issue. If you have variant allele frequencies of lower than 50% in your tumor, doesn't that mean that half your tumor is not going to be targeted by your T cell population you're infusing, and you're going to miss half the tumor? Why do you expect a low allele frequency to be acceptable here?
Yeah, well, I didn't mean to suggest that a low allele frequency was acceptable. I didn't, just because of the limited time, I didn't go into the frequency of the mutations that are recognized by T cells. But if you actually go back and look at the data, in most cases, those antigens are fairly frequent in terms of and I would say even something that's 25-30% variant allele is statistically, it's probably hard to distinguish that from 50%. So I would say, really, we probably only have to worry about things that are in the 10 low-digit range in terms of variant allele frequencies. For the most part, I don't think that our TIL generally recognize those. So, I mean, that's something we just need to determine, right? I mean, just by studying more and more patients. And so, whether or not that's going to be true in the future, when we study more patients, I don't know. But I guess I would argue that the patient, certainly that had 10 mutations that were recognized, in a sense, if you have subclonal populations, since we can't determine, if those mutations are all present on the same cell unless we do single cell analysis, 50% of the cells express one mutation and 50% of the cells express the other, we're still going to be able to target them. So, yeah.
So this is an extraordinary talk. You have me convinced that this is the way forward to manage heterogeneity. I think we don't really need our clinical trial session next. This is the answer, without a doubt, in my view. It's the future. It's going to, I think, trump all targeted therapy approaches by a country mile. But that aside, I just wonder, from colorectal cancer as a disease model, it's quite an exciting model to study, because you've got microsatellite instability, which accounts for about 5% of patients with metastatic disease, and then a large cohort of patients with chromosomal instability. And you mentioned that colorectal cancer does not seem to be particularly sensitive or sensitive at all to immunotherapy-based approaches. And I'm not an immunologist, but one thing that's always intrigued me is whether chromosomal instability might be a way of generating diversity by stealth. So you alter gene dosage, but you don't actually generate neoantigens that allow a T cell response to be initiated. And I wondered if you'd actually treated any patients with microsatellite unstable colorectal cancer that have hypermutations, and see if you see responses in that group particularly, albeit the small 5% of patients with metastatic disease.
Yeah, I mean, we have one patient with Lynch syndrome, and that patient does have a very large number of non-synonymous mutations. We haven't I don't believe that we've looked at that patient. We just recently got exome data from that patient. So it'd be interesting to see if that high mutational burden shows what we see in melanoma, namely that there's a correlation between the number of mutations and the number of antigen targets. But we just haven't. And that patient didn't receive treatment, I don't believe. So we don't have an answer to that question.
In terms of, I'm not an immunologist, but can the immune system distinguish alterations in gene dosage?
Okay, I could talk a long time about that. Basically, when we started cloning tumor antigens decades ago, we assumed that some of the targets would be overexpressed transcripts.
Yeah.
And it's been remarkable how few of the products that are recognized by the immune system are just simply overexpressed, at least by T cells. There are a few examples, and we've cloned or identified a few of them. But really, when you look at melanocyte antigens, they're there at extremely high levels in the normal tissue. So that's not overexpression. I mean, and even things like CEA, people talk about CEA expression. The cells in the crypt that are CEA positive are very, very strongly positive. They express extremely high levels. There's not a lot of cells. Just at the same time, there's not a lot of melanocytes in the skin. But the levels of expression are very, very high. The cancer germline antigens like MAGE and ESO are basically turned off in the adult. They're not expressed in any normal adult tissues with the exception of the testes. And those cells in the testes don't express MHC molecules. So when you actually look at it, it's actually kind of surprising. It's not what we expected when we first looked at that. I mean, there's serological antigens that are generated by overexpression, but no, the T cells are pretty exquisitely sensitive to very, very small levels of antigen expression. So that's a pretty rare thing, it turns out. So yeah.
I also have a question that, because I'm not an immunologist and I don't really understand how this works, so it's probably a totally ignorant question, but to what extent can you get the response to switch to the things you weren't targeting? That is, if the T cells are coming in and are recognizing a particular target, do you ever get, and maybe you're knocking down that clone, do you ever get new T cells that are responding to other mutated antigens that are also in the environment? Does that make sense?
Yeah, I mean, you're talking about antigen spreading, which is a hot topic, and a lot of people, some people have shown examples of antigen spreading. In our TIL trials, actually, Ton Schumacher, who developed the Tetramer approach, has examples of both. Has examples of where, in the TIL patients, he's examined the specificity of the cells we infused and the specificity of the cells that are present after transfer. And they line up almost perfectly. But there may be some evidence in the checkpoint blockade trials where there are new specificities that arise. So I think you have both situations. In that case, of course, you're not ablating the patient, though, right? Whereas we're ablating the patient. So presumably, there may be less of a chance for that kind of antigen spreading. Although eventually, the patients' own cells will come back.
And that's the basis of the hope that the adaptive immune system can adapt to the adapting tumor, right?
Right.
Chase it down.
Right. Yeah, we just don't know. I haven't studied that.
I think we'll take our break now. Thank you very much.
Okay, great. Thanks.